Hello friends, welcome to GRADEUP. Welcome to another class of EPFO General Science and Technology. So over the past few classes, we have finished the most important parts of physics and chemistry, the basic understanding which is required for competitive examinations. One second. Yeah. And in today's class, we are going to begin with the biology part. Okay, right from the what is a cell, what is a cell structure, what are the components of cell, what is DNA, RNA, right from such a basic level to the more advanced levels of, the genetics or the parts of the human body or anatomy of human body, immunity, diseases, everything will be taken up. Okay, this is the last part of our general science discussion. So let us see the first part of the discussion of biology. Typically any class of biology begins with this only, which is actually the cell structure. What exactly is a cell and what is the structure of the cell? So the cell, it was first time discovered by Robert Hooke in 1665. So way back in the 17th century itself he had discovered. He had observed the slices of cork. Okay, cork, जो wine bottle के ऊपर होता है, that thick cork one, by the simple magnifying device. So you get it from the bark of a tree. So he took the slices of the cork and he observed it under the microscope. So he had observed something like cells, but these are the dead cells, obviously. Cork is dead cells. Okay, so he had found some sort of fundamental structure which are there inside the cell. Sorry. Okay. Similarly, later Anton von Leeuwenhoek, he was the first to discover the living cells. Okay, he had seen the cells inside that microscope, and because they were sort of living, they had that contracting and expanding sort of cell movements, he called them animolecules. Okay, all this actually are there in your NCRTs. So the story of the discovery of cell, first time Robert Hooke, later Anton von Leeuwenhoek, they can be asked. Direct question, who was the first scientist who had experimented with the cell structure or who had seen? It is Leeuwenhoek. Sorry, it is your Robert Hooke. So cells are the fundamental structural units of living. All units of life actually begin from cell. And in the living world, there are different organisms. So there are different types of cells. We have broadly two types of organisms. One is the multicellular and other is unicellular organisms. So unicellular are the organisms which are only one cell, single cell. Okay, like your amoeba, Chlamydomonas, Paramecium, bacteria. These are the single cellular organisms. And the multicellular are the ones which are fungi, plants, animals. Okay, not only frogs but all animals. We are all multicellular organisms. Now please note this is different from prokaryotic and eukaryotic. If generally my students, they get confused that prokaryotic is unicellular, eukaryotic. It's not at all. Prokaryotic is the it is when nucleus and other cell organelles, if they are there in a correct way, then it is eukaryotic. If the nucleus and all is not there properly, it is prokaryotic. Okay, we'll be discussing that. But here, don't get confused. Here, unicellular is one cell, multicellular is multiple cells. So, multicellular organisms, there are billions of cells which start with their life cycle, and the fertilized egg actually multiplies into different cells and the organism develops. Actually, roughly, if you see the cells, they develop to tissues, and the tissues next they develop into your organs. So that's why cells are the building blocks of any organism. So single cell organism, they perform all the necessary functions of ingestion, digestion of food, respiration, excretion. Hey, ek hi cell, but it takes care of the entire life process of that organism. And the main difference between single cell perform all the functions of unicellular and multicellular is there is a specialized group of cells in the multicellular. As I told, that specialized group of cells form the tissues and that form the organs. So if we see the unicellular organisms, the cells are round, spherical or elongated. Sometimes they become long, pointed, different shapes are possible. And some are even branched, like your neurons. In the central nervous system, we have the neurons with the nodes of Ranbir, with the synapses and all. So that is in a long shape. So through this class, whatever I discuss the cell structure and all, it is for the normal body cells, which I'm talking about, in the normal cytology. But actually, there are different types of cells. Amoeba, as you know, it's a single-celled organism. It has a definite shape, but its shape keeps on changing by this movement. Okay, it has this pseudopods. Pseudo means false, pod means feet. So it's basically false feet. So they're not real feet, they're like the false feet, and they help in movement of amoeba. And when there is a food particle, for example, this pseudopods, they capture that food particle and they digest by something called as phagocytosis. So pseudopods are important for transportation and also for the phagocytosis. Okay, this class actually it's descriptive, more than logic or reasoning. Actually, most of biology is like that. You should just remember the terms schematically. Without any doubts, you should remember the terms, then you can answer any question on biology. And cells, as I've told, there are different shapes. Similarly, the different sizes. They can be small, up to micrometer or micron length. The smallest cells are there in the bacteria, which are 0.1 to 0.5 micrometer. And the largest cell is your egg of ostrich. Egg basically is one cell. So that egg of ostrich is the largest. It is 17 into 13 centimeters. So about this big, the egg of an ostrich. And the size of the cell has no relation with the size of the body of animal or the plant. So it's the basic structural unit. It forms tissues and they form the organs. We have discussed all this. Now, what are the parts of the cell? So initially, I'll show you the diagram also, but initially you should be knowing that this cell, it has the cell membrane, which is also called plasma membrane, which is the outer covering of a cell wall, and inside there is a nucleus. And between the nucleus and your cell membrane, we have the liquid surrounding the nucleus, which is called as cytoplasm. Okay, and it's on the cytoplasm that different cell organelles actually exist. And nucleus and cytoplasm together, they are called as protoplasm. Okay, basically it consists of all the living organisms inside the cell, the protoplasm. So the cell membrane, as I told, it separates the cell from one another, and they're made up of lipids, proteins, and carbohydrate chains. Okay, lipids are basically your fats. So fats, proteins, and carbohydrate chains. We'll be discussing actually what are fats, what are proteins, what are carbohydrates. In the nutrition chapter, we'll be discussing. So based on all these, the cell membrane is made and it is porous. It is porous and selectively permeable. Okay, just like your, say, watchman or security guard, it is selectively permeable. That means it allows only some organisms or some substances to enter the cell. Not all the organisms, all the substances, but only some substances are allowed by this method of osmosis. So osmosis, I hope you know, it is the change, it is the transport from higher concentration to lower concentration. For example, we have the say let us take a cell. Let us take a cell and in the respiration that will make it clear for you. In the cellular respiration, the cells respire, the mitochondria produces energy, and there is a lot of carbon dioxide which is accumulated in the cell. Okay, there is some oxygen also, and the blood, as it reaches the cells, the blood has something called as hemoglobin. And on the hemoglobin, we have the oxygen molecules. One second. So as the concentration of the oxygen molecules in the cell decreases and carbon dioxide concentration increases, there is some osmosis. So the carbon dioxide is excluded from the cell and the oxygen which is there in the blood, it enters the cell, okay, at the respiration level. So in this way, at the cell membrane level, there is osmosis. So there is exchange of the gases or exchange of any substances actually. So that is your cell membrane. And we have the cell cytoplasm. Cytoplasm is the jelly-like substance which is there between the cell membrane and nucleus, as I showed you. And in the cytoplasm itself, we have different organelles, like our mitochondria, lysosomes, and so on. We'll be discussing in today's class. All of them, they float here in the cytoplasm. And additionally, it's not there in animal cells, but in plant cells, there is additionally something called a cell wall in the plant cells. So it gives shape and rigidity to the cells because it's made up of cellulose. So, because of this rigid structure, when there is a heavy wind or, heavy water flowing or high temperature, in all those situations, the plant can remain strong. It can get that rigidity or turgidity because of this cytoplasm, because of this cell wall. So when a living plant cell, it loses water through osmosis, there is shrinkage, and this is called as plasmolysis. Fine. And similarly, we have the nucleus. Nucleus is a spherical, it is located in the center of cell. It is separated by nuclear membrane. So on the nucleus also, there is something called as nuclear membrane. Okay, so far so good, just basic stuff. And what does nucleus do? It uses the information in something called as chromosomes. And these chromosomes are the ones which decide how an organelle should function, how the cell should function, and on the whole, how the organism itself can function. Okay. And nucleus also has some concentrated material called nucleolus, which carry the genes. Okay, genes are the ones which help in inheritance or transfer of characteristics. And these genes are actually consisting of the DNA. Okay. I'll tell you all of this in depth. We'll know what is DNA, what is RNA, and all. So DNA is the actual genetic matter. The meaningful sequences of DNA is your genes. And this DNA itself, it is composed to form your chromosomes. It's bound in chromatin, but on the whole, it is chromosomes. Okay, so the chromosomes, they carry information of inheritance from parents to the next generation in the form of this DNA molecules. And also we have something called as vacuoles. Vacuoles are the storage bubbles which are found in the cell. They store the food and all the nutrients which the cell requires. And in plant cells, vacuoles are very big. Vacuoles are much larger than the animal cells, and they are very big, so they'll also help in giving the rigidity of the plant cells. And also plant cells have the tiny colored structures called as plastids. So plastids also, I'll tell towards the end of the class that there are different plastids. They basically give the color. Okay, they carry the pigments of various colors like orange, red, green, or even colorless. Like for example, these orange, red, green which we have, they are the chromoplasts. And this colorless ones are your leucoplasts. And the green ones are obviously our chloroplasts. So they are the ones which give the color to the plant. You must be thinking, sir, red, blue, green, where do we have? This red, blue, orange are basically for the fruits or other parts of the flower. If there's a red stem, that is because of the chromoplasts. And as I told you in the beginning, please don't get confused between a prokaryote and a unicellular. Both are different. Prokaryote is something in which the nucleus is not well defined. Okay, and thus nucleus is called as nucleoid in that prokaryotic cell. So there is a single circular chromosome in this prokaryotic cells. There are no membrane bound organelles, and in the eukaryote ones there is a proper nucleus it has a linear chromosome and there is membrane bound cell organelles. For example, you can see this prokaryotic cell, say maybe a bacterial cell. It has, you can see, simple, see how is the DNA? DNA is there right in the cell. There is no proper nucleus, and in that DNA is not there. It is there right in the cell. Ribosomes are floating, your fimbriae are there floating, but it also has a cell wall and plasma membrane. Okay, this is a prokaryotic cell. Eukaryotic cell are our cells which we have. It has a nice little nucleus. Okay, and this is the nucleus. It has a nice nucleus, and inside the nucleus there is a nucleolus, there is a DNA, RNA, there is the endoplasmic reticulum, there are ribosomes on that, there is the chromatin of the DNA, then there is Golgi apparatus, there is nuclear envelope, vacuoles are there, ribosomes are there, cytoplasm is there, plasma membrane, DNA, all of this they are there. Okay, so it is well bounded. Like, see, even the nucleus has a membrane. This vacuoles, ribosomes, which you're seeing, even they are having membranes. So they're the membrane bound, these eukaryotic cells. Prokaryotic cells are not like this. So if we compare the plant and animal cells, roughly I would say that the plant cells have a cell wall. Okay, in animal cells there is no cell wall. And they have chloroplasts. The animals, they don't have chloroplasts. They have something called as centrioles, which we'll see. And rest, most of them are similar. And in the plant cells, there is a big vacuole. You see, this is one big vacuole they have. Okay, and these vacuoles also tend to act as lysosomes. They are called as lytic vacuoles actually. Okay, these type of plant and animal cells comparison we can make. So just try to answer this question. I'll tell you more about cell membrane and walls. This was asked in the prelims 2020, UPSC prelims 2020, UPSC CSC prelims 2020. This question was asked just recently on October. Try to answer this question. Anyone? We just discussed plant cells, animal cells. Pratik is saying C. Any other answers? Divya also is saying C. Very good. So as we have discussed, the plant cells have cell walls while animal cells do not. So this is correct. The plant cells do not have plasma membrane unlike animal cell walls which do. Is this correct? No. Plant cells also do have the plasma membrane like animal cells. Okay, animal cells and plant cells both have this plasma membrane or cell membrane. And the mature plant cell has one large vacuole while animal cell has many small vacuoles. This is correct. Okay, as I've showed, it has one large vacuole, this plant cell. So that is a brief difference between both of them. So the answer is C, 1 and 3 only. So such basic questions from what is cell, from the cell structure also they have asked in UPSC, which can be also expected for our EPFO exam. But for our EPFO exam, this multiple bits may not be asked, because for a EPFO, there is more stress on speed rather than, this sort of in-depth. You should be faster, since there are more subjects and more questions which are asked. So they might give you just one option questions. So cell membrane, as I have told you, it is there in both plants and animals. It's made up of proteins and lipids. It is semi-permeable in nature. So it helps in the transportation of the molecules. Cell wall was discovered by Robert Hooke itself when he had discovered the cells, and they are the non-living structure. They're giving the shape to the cell. They are made up of cellulose. I discussed that they'll help give the rigidity to the plant cells in the wind, against the water and the other conditions. It gives a rigidity. Fine. So both plant and animal cells have cell membrane, cytoplasm, nucleus, nuclear membrane, but plants have larger vacuoles. Plants, why do they have larger vacuoles? One of the important thing is they cannot move. Plant, like animals, we can move. We can, go to maybe say ice cream parlor or go to some pizza place. We can eat food, but your plants cannot move. So whatever food they're getting at that point, if there is more rain or from the soil, whatever food they're getting, that food they'll have to store in their cell structure for a longer time. So that's why they have the bigger vacuoles. And they also have plastids, and they have the cell walls. So please remember this slide. It's very important. I have tried to summarize here. What are the differences between plant cell and animal cells? Okay, and see, this is my teaching material. This exact material you won't be getting in your notes. Okay, I've added a lot of stuff to make it comprehensive. So you can either make notes of this, or you can take screenshots of these classes, especially bio classes. Physics and chemistry classes are more or less the notes which you got, you must have seen. But for bio, I had to add a lot of extra things to make it comprehensive, to make sure that, we'll get all the bio questions correct. So nucleus, we have seen nucleus is the main central structure which has the genetic material. It was discovered by Robert Brown in 1831. It is the brain of the cell. It is in the center of the cell. It has DNA, chromosomes, and nucleolus. So you can see if this is the nucleus, inside the nucleus we have this chromosomes which have the DNA. Okay, chromosomes are the ones which are made of DNA. All this we have DNA in between them. And we also have the nucleolus at the center of it. And most important, we have the DNA. So let us see what is DNA. So DNA, it contains genetic information. It is replicated during cell division. So DNA, it's made up of 3 components. Okay, initially 3 components. It has a phosphate group, pentose sugar, and most importantly, it has the nitrogenous bases. There are 5 nitrogenous bases. In DNA, we have the adenine, thymine, cytosine, and guanine. In RNA, we have the adenine, uracil, cytosine, and guanine. Okay, so they are composed of these nitrogenous bases. Mostly, the DNA structure is composed of this nitrogenous bases, and additionally, they have a pentose sugar and phosphate group. Okay, you can see, for example, in this figure, can you see here we have the nitrogenous bases. These are the nitrogenous bases. So we have a pentose sugar and a phosphate group to it. And those containing your deoxyribose sugar is DNA, and the ribose sugar is RNA, which means you can see here. Okay, if this is the DNA structure, these are your nitrogenous bases. Say this is A, this is G. Okay, they have given color. This is T, yellow is adenine A, guanine is G, cytosine is blue, Okay, so in this sort of sequence, and always adenine has a hydrogen bond with thymine, and your cytosine has with guanine. And in RNA, same thing there is there, but instead of your T, instead of thymine, we have the uracil. Okay, and here there is the double-stranded sugar phosphate to the nitrogenous base. This sugar phosphate you can see here is the structure of the sugar phosphate is like this. So main difference between DNA RNA also is that here there is no oxygen. Here, okay, here you can see there is one oxygen. So there is a deoxyribose. This is ribose. This is the formula of ribose sugar. So in this, if I am removing one oxygen, I get DNA. Okay, this is ribose sugar. So deoxy, matlab in one ribose sugar, I am removing one oxygen. So in this ribose sugar, I am removing one oxygen. I get this. This is my deoxyribose. Okay, that phosphate group basically. So that phosphate group without the oxygen is my deoxyribose nucleic acid. So the only difference between two is there is one oxygen less in the deoxyribose. And most organisms, we have DNA as our genetic material in our nucleus, but some viruses also have RNA as genetic material. Can somebody tell me the name of the famous virus which has RNA as genetic material? Anyone? Tell me one famous virus which has RNA as its genetic material. You know this. I'm 100% sure you know this. Anyone? Nobody knows one example of RNA virus? Yes, coronavirus. Okay. COVID-19 coronavirus, which is there, it is also an RNA virus. Okay, that's why it actually changes, it keeps changing its structure very fast. That's why there are different strains. That's because it's an RNA virus. Similarly, tobacco, mosaic virus, QB bacteriophage, COVID-19. Actually, the whole SARS, not only your COVID-19, but SARS virus, MERS, Middle East Respiratory Syndrome. So all these viruses are your RNA virus. Okay. Now, since all the living organisms, they have DNA, you can mix and match them. That is the reason why you can clone, you can create hybrids. Because in me, in you, in a cat, in a horse, in an elephant, in a giraffe, in all the animals, we have this only, ATCG. Okay, it's just the sequence how it is changed and the ordering of it. All of us, for example, you and me, we have 99.9% similar ATCG in our structure. But my eye shape, my hair color, my hair shape, my height, my weight, all of this is dependent on the 0.1% changes in our DNA structures. So they are joined by hydrogen bonds, as I've told, and I also told you this, adenine bonds with thymine and cytosine bonds with guanine. And in the RNA, instead of thymine, we have uracil. And the structure, if you see, it is somewhat like a ladder. Okay, it is like this, so they bond like this in the DNA structure. Okay, same thing. One important thing you should know is not all DNA is there in our nucleus. Some amount of DNA is there even in the mitochondria, where it is called as mitochondrial DNA. Okay, a very small amount is there even in the mitochondria. Okay, so this is a monomer of, we have seen yesterday, monomers and polymers, we have seen the difference. So this DNA is a long polymer of the deoxyribonucleotides. This is the monomer. So this keeps on repeating to form you the DNA. And the length of DNA is usually length of nucleotides, fine. Similarly, what is RNA? RNA is your ribonucleic. It has that extra oxygen. It has a long chain of nucleotides. So what is a nucleotide? Nucleotide is a nitrogen base, the sugar, and the phosphate. Okay, but most importantly, nitrogenous bases are the most important. If you know this nitrogen base, it's important. It's enough. And this is DNA is double-stranded, but RNA is a single-stranded molecule. Okay, you can see here it is just one strand, single-stranded molecule. DNA we have seen, it contains the main genetic material of a body. It's important for the DNA. It is the one which is responsible for our eyes, hair color, all our characteristics, how we behave, our functional, our structural components, everything is there with DNA. What does RNA do? We have seen in some viruses, they carry the genetic material, like the COVID-19 virus. And the main job of RNA is to transfer the genetic code, which is required for creation of proteins from nucleus to ribosome. I'll tell you what is nucleus, what is ribosome, we'll discuss. But from a nucleus to ribosome, the RNA is the one which carries. Okay, like specifically it's called messenger RNA. So this prevents the DNA from leaving the nucleus, because DNA is there somewhere in the nucleus and the message has to be created to the ribosome. Ribosome is where the proteins are created. So the RNA is the one which transfers the message. So they keep the DNA and genetic code from damage, and without the RNA, the proteins can never be made. So some RNAs also act as enzymes, like some RNA, they are called as ribozymes, and they exhibit features of an enzyme. What are enzymes? We'll be discussing. They are the ones which fasten a biological process. So RNA can also act as enzymes. Okay, and some basic components, some basic understanding you should know. The main functioning of genes, it is revealed in the dogma that DNA is the one which is used to synthesize RNA, and RNA later undergoes translation to produce proteins. So DNA, it converts to RNA by transcription. And RNA, as I've told, it codes for proteins by translation. This is the central dogma of the DNA functioning. So this is how the DNA functions to create proteins in our body. Because all the eye color, hair color, all the height, everything, ultimately it's proteins. It's nothing but proteins. So this DNA, it creates proteins in this way. Okay. Now I want to ask you one question. Can somebody tell me the full form of RT-PCR? RT-PCR, some of you must have got the test also in the COVID-19. What is RT-PCR test? Where they take that nasopharyngeal swab and they test for the if you have COVID-19 or not. Somebody tell me the full form of RT-PCR. I hope you are reading current affairs. Everyone is reading current affairs for EPFO. What is the full form of RT-PCR? Nobody knows what is RT-PCR. Divya? No? RT-PCR is reverse transcriptase polymerase chain reaction. So because your COVID-19 is an RNA virus, this is COVID-19. So I have to convert it into DNA so that I can make something called as PCR. PCR is where one small DNA particle is created into multiple. It is cloned into different DNA. So from one DNA molecule, I can create multiple DNAs by this PCR mechanism. But COVID-19 is this RNA virus. How can I convert it to DNA? That is how I'll make it by reverse transcriptase. So this is transcription. I'm converting RNA to DNA by reverse transcription. Okay, that is how its name came, RT-PCR. So actually many people who took this RT-PCR test who found out, yes, correct Divya, but you've been a bit late. So that is the reverse transcription. That is how they are making. The RNA, which is COVID-19 virus, which is RNA, they are reverse transcriptase it to convert into DNA, and from the DNA they are doing PCR to get the multiple copies of DNA, and ultimately they'll test if you have COVID-19 or not. And this DNA, they are packaged into thread-like structures called as chromosomes. So you can see this is the DNA structure as we have seen. It is bound onto this chromosome. It is bound onto this histones. Okay, these are your histones. It is bound onto the histones and they form the chromatin fiber. So the chromatin fiber ultimately forms the chromosomes in a person. So each chromosome is made up of a DNA tightly coiled many times around histones which support the structure. Okay, so that is my DNA and the chromosomes. Now what is gene? So DNA we have seen there are different sequences, like say this is a DNA sequence. Okay, big DNA sequence we have. Now say this sequence, this GG8ATA is the one which is determining your hair color. Okay, take this ATCG. Maybe this is determining your height. So what is this? This is a sequence of your DNA which is meaningful. Okay, so the sequence of nitrogenous bases or DNA which is forming a meaningful sequence is my gene. So this is a gene. Of course, this is not a gene for hair color. I'm just hypothetically writing. But in this big sequence, there are roughly 3.2 billion base pairs in your body. So in that, some base pairs, they code for a specific gene. Okay, specific hair color or specific intelligence, specific eye color, specific emotions if you have, that is also coded in the form of genes. So everything is coded into your genes. And genetics is the branch of biology which studies this heredity and variations. So how characteristics they pass on from the parent to the offspring, it is studied in genetics. So gene which is inherited, it is a biological character, and sometimes there can be a variation. Of course, your child is not exactly like parents. So there is a variation, there is a mutation. So how it is inherited and how the gene expression is seen as proteins is the main basis of the variability. This is your DNA. This DNA will code for RNA and RNA will form the proteins. Okay, so this is happening in the parents, but what is happening in parents need not happen in child. So there is some variability also. That is also studied in genetics. Okay, and part of your current affairs, you should be knowing that the Indian government is undertaking something called as Genome India project. So this is conducted by the Department of Biotechnology. 20 leading institutes, including IITs, IISc, Bangalore, and few IITs, they are taking up this. And main nodal point is by the Center for Brain Research. It is in IISc, Bangalore. So what are they doing? They are collecting the samples of 10,000 persons to understand the Indians. In the Indians, how the DNA structure is there. Okay, from 10,000 persons, they are analyzing this 3.2 billion base pairs, and they want to know which gene is coding for what, which gene is coding for which function. So it's a very ambitious project which we are taking up. Yes, are we back? What was the last part which you saw? I think you have seen this 10,000. Yeah. So under the Genome India project, in the Indian government is taking 10,000 people, 10,000 specimens, and we are conducting and we are trying to understand the genome structure of these 10,000 people. Because earlier in the Western countries, mainly led by the USA, they had conducted the Human Genome Project in 1990s. So by that they had analyzed many people from the Human Genome Project, and they could get this human genome. They could get that what are the genes, which genes are coding for what proteins. But the problem with Human Genome Project was that most of the people in that Human Genome Project were Caucasian. That means they were white people. But in India, of course, we don't have, we have a Caucasian element also, but we have many other races. We have the Caucasian race, we have the Negrito race, we have the Mongoloid race. So different race mixtures we have in India. And they had considered in the Human Genome Project mainly urban middle class samples. So it is not representative of all human beings. So that's why the Indian government is taking up this Human Genome Project. Because in India, we have a vast mixing of races. There is a vast diversity of races in the horizontal level and the vertical level. Because as I told, the Negrito, Caucasian and your Mongoloid races are there admixtured in India. And there is a vertical similarity. That means in our India, we have the caste endogamy. We have marriages within the caste and marriages within the same religion. So that genetic structure which is there, that has been preserved in India. So because of this reasons, we are having our own sort of study, which is a Genome India project. Okay, very important. It can be asked in the exam. Please note. By the Genome India project, we want to know the Indian genome structure, the Indian genomic data. So it will help us develop an indigenous capacity to generate, maintain, analyze, utilize, and communicate a large-scale genome data in a scalable manner. Fine. I hope it's clear. In Genome India project, I hope every one of you understood. Okay, so that is the whole aspect of nucleus. What is DNA, what is RNA? We have seen comprehensively, you have seen. Okay, at such a depth in books also you won't find. I sourced from various material and I prepared this for you. Okay, now let us get back to the cell structure. We have seen that the nucleus has DNA and chromosome and other structures. There is also something called as nucleolus, which I have told you. It occupies 25% of the volume of nucleus. There is no chromosome, but it has RNA. Okay, most DNA, RNA, DNA and chromosome which is there, they are there in the nucleus itself. Okay, here for example, we have the chromosomes like this. But there is one part which is called as nucleolus, which is the granular component, which has a granular component, the ribosome assembly, fibrillar component, the rRNA. Okay, different components are there. Just remember nucleolus, there is a RNA, more RNA is there. Similarly, we have ribosomes. Ribosomes are the ones which are the protein factory of the cell. This is where the proteins are made. They are also present in endoplasmic reticulum and mitochondria. They also have the RNA. Okay, actually, let me show you a more detailed description. 1 minute, give me 1 minute. Yes. Let us see the cell organelles. So we have seen nucleus, cytoplasm, and DNA, RNA. Now we should know some important cell organelles. Like first we have is, I was telling you about ribosomes. So ribosomes which are present in the active cells, they are the ones which are responsible for protein manufacture, and they send the protein manufacture to this endoplasmic reticulum. Okay, on the endoplasmic reticulum, we have the ribosomes which actually code for proteins, and these proteins manufacture which are there, it is coded, it travels through the endoplasmic reticulum, and this is directly connected to your nucleus, to the nuclear envelope. And similarly, we also have a smooth endoplasmic reticulum here, which is used for the manufacture of fat molecules, lipids, and other components of cell function. And they are also used in the detoxification, like in liver, and this process is called as membrane biogenesis. That means they help in the building of the cell membrane. So naam mein hai membrane biogenesis. That is where the smooth endoplasmic reticulum comes into use. I'll just show you the, okay, I can't keep showing you and not showing the structure. Okay, just remember the structure. I'll be explaining them one by one. I already showed you this. We have the nucleus. Nucleus में we have the DNA. उसके अंदर we have nucleolus जिसमें RNA है. And attached to the nucleus, as you can see, is this endoplasmic reticulum. This is endoplasmic reticulum. Okay. On the endoplasmic reticulum are the ribosomes. Okay, this is the rough endoplasmic reticulum, and similarly we have Golgi apparatus, which is mainly involved in the manufacturing and packaging of the proteins which are there. Okay, I'll tell you about Golgi apparatus. Similarly, we have cytoplasms. We have our where is lysosome? Lysosome is not there. There is also one lysosome, which are the suicide bags of the cell. There is a mitochondria. Mitochondria is where the ATP is made. And as I told you before, in the mitochondria also some DNA is there. Okay, so I hope you remember this picture. Just go through this picture for 1 minute. Then, so that when I explain you all the cell structures, it will be clear to you. Just see this for 1 minute. Okay. So endoplasmic reticulum, I told you, they have those long tubules called as oblong bags, and there are two endoplasmic reticulums. One is your rough endoplasmic reticulum, which has the ribosomes on its walls, and the smooth endoplasmic reticulum. Similarly, we have a Golgi apparatus. Remember, in the Golgi apparatus, it was there, like this sort of structures. So they are mainly involved for the packaging. The material synthesized in ER, it is packaged. Just like in an industry, if you are making a cotton textile, that cloth fabric which you are making, you are packaging it, or packaging go down also is there. That happens in this Golgi apparatus. So it is involved in storage, modification, and packaging in the vesicles, and it's also involved in formation of lysosomes. What are lysosomes? Lysosomes are the waste disposable system of the cell. So they are the ones which keep the cell clean by digesting any foreign material, and it has the digestive enzymes, and any waste material which are there, it is disposed in the lysosomes. And it's also called as suicide bag of the cell, because when a cell gets damaged, the lysosome might burst, and the enzyme which is there, it digests the own cell. So if there is a lot of stress, even in your body, when you're going through a lot of stress, 80s and 70s is the size of the ribosomes. Okay, there we can, there these organisms are the ones which commit a suicide, so the cell gets destroyed, which is lysosome. Similarly, mitochondria also, I showed you, mitochondria are the powerhouse of the cell. So all the energy which is required for the chemical activities, it is done by mitochondria, and it has the ATP molecules. So ATP is the energy currency, like how we have joules, how we have, you know, different units of energy. It is measured in ATP in a cell level. So all the body energy, for example, if you're running, if you're walking, if you're studying, or I'm talking presently, I'm using up ATP in my body, and it is used in the mitochondria. And it has two membrane coverings. Outer membrane is porous, inner is deeply folded, something like this. And they have their own DNA and ribosomes. So because they're having their own DNA and ribosomes, they can make their own proteins. Okay. So mitochondria, they're actually very strange. Fine. I hope you understand. DNA is there only in the nucleus. If there is a statement, please get it wrong. Okay, DNA is also there in the mitochondria. Please remember this point. And the last we have are plastids. Plastids, as I told you, they're there only in the plant walls, plant cell walls. There are chromoplasts, leucoplasts, which are white or colorless. I told you in the beginning. And the ones which have this green colored pigment are your chloroplast. This is the structure of the chloroplast. So the green color of the plant is given by the chloroplast. And there are different other yellow and orange pigments we have in our chromoplasts. And the internal organization of the plastids consists of numerous membrane layers which are embedded in a material called stroma. In this material, they are embedded. Okay, this thylakoid membranes are embedded. Fine. And the plastids also have their own DNA and ribosomes. Okay, just like in human body we don't have plastids, but in plant cell walls they have their own plastids, and the plastids have their own DNA. Okay, these plastids were discovered by Ernst Haeckel. Ernst Haeckel, I hope you know Ernst Haeckel. He was the one who had coined the term ecology. Okay, he's the father of ecology. So he discovered these plastids. They are present in the plant cell wall, and you should remember these three. They are asked in the exams. What is chloroplast, what is chromoplast, and what is leucoplast? Okay, chloroplast has chlorophyll, which absorbs the sunlight. Chromoplast has carotenoids, which is red, orange, yellow color. They are found in the flowers and fruits. Leucoplasts have no pigment. They are used to store starch. They are mainly for the storage purposes. So this type of one-liners are asked for our exam. We are not biology students. We are not studying for NEET or other exams. Just one-liners we should be knowing. For example, which cell organelle is called as the suicide bag of the cell? Tell me the name of the organelle. कौन सा organelle? It's called suicide bag. When the cell comes under any pressure, it degenerates and it bursts. Yes, very good, Divya. Lysosome. Similarly, in which organelle is the packaging done? Packaging of the protein, packaging of the cellular substances. Yes, not Golgi bodies, Golgi apparatus. Similarly, which is the powerhouse of the cell? This is very famous in many competitive exams is asked. Powerhouse of cell क्या है? Yes, very good. Mitochondria. So this type of one-liners are asked. No need of reading them in depth. What is covered in this class is more than enough. Okay, you can go through your notes, and if you're interested, if you find this interesting and you want to have a comprehensive understanding, more in-depth understanding, because we have just done for 1 hour, you can read the NCERT class 8th and 9th, just two chapters. Just two chapters. In the class 8, they have given about nucleus and DNA. In the class 9, they tell you about the cell organelles. Okay, with more diagrams, what are the more functions. I have told you for the competitive exams, but if you're interested of learning them in depth, you can see. In the 9th class especially, this was one chapter. Cell and its structure, you can read it. Okay. So that's it, friends. That's the end of today's class. Let's meet tomorrow for classification. Okay, animal kingdom classification will begin tomorrow. Okay, any doubts in this class, you can mail me to b.teja@gradeup.co. Okay, so thank you so much. Let's meet tomorrow, same time, 4 o'clock. Okay, bye.